Good morning, everyone. I'm Dr. Romana Mirte, and I welcome you all to this online training program organized by MycoAsia Journals. Today, we are having a very eminent personality with us, Dr. Belle Shenoy. He'll conduct training program on phylogenetic analysis using Mega software. I welcome you, sir. I request Akshata to introduce today's speaker. Akshata, it's over to you.
Thank you, Romana. It's my immense pleasure to introduce Dr. Belle Damodara Shenoy, sir, today's speaker. Dr. Belle Damodara Shenoy is a fungal taxonomist by training. After completing his bachelor's and master's degree from India, he pursued his PhD in fungal taxonomy at the University of Hong Kong under the guidance of Professor Kevin Hyde and Dr. Rajesh Jivan. His PhD thesis, titled Multigene Phylogeny of the Selected Anamorphic Ascomycetes, which deals with the evolutionary relationships of an asexual genera such as Sporidesmium complex and provided insights into the implications of their polyphily in the fungal taxonomy. Dr. Shenoy, for a brief period of time, he worked as a postdoctoral fellow with Dr. Keith Siffert at the Agriculture and Agri-Food Canada, Ottawa, on DNA barcoding of Fusarium. In 2008, Dr. Shenoy returned to India and joined the microbial type of culture collection, CSIR Institute of Microbial Technology, Chandigarh, CSIR Impact. He supervised two PhD students at CSIR Impact and published several high-impact papers on fungal taxonomy, phylogeny, DNA barcoding, and comparative genomics. In 2013, Dr. Shenoy moved to the CSIR National Institute of Oceanography, Goa, and initiated the research on diversity and taxonomy of microbes associated with the tar balls. He is presently working at CSIR NIO Regional Center, Visakhapatnam, Andhra Pradesh, India. Dr. Shenoy has so far published 45 papers in reputed international journals. He is a founding member and presently the managing editor of a new mycology journal, MycoAsia, the Journal of Modern Mycology. Dr. Shenoy has been a member of the International Subcommission on Coelotrichum taxonomy. Dr. Shenoy is on the advisory board of Amity Institute of Oceanography and the Atmospheric Sciences India. His non-scientific hobbies includes traveling and exploring India and writing the poems. Over to you, sir.
Thank you, Akshata, for your kind introduction of me to today's audience. Good morning, ladies and gentlemen. Am I audible? Okay, ladies and gentlemen, today we'll be spending some time together. We'll be talking about phylogenetic analysis, the principles behind phylogenetic analysis, then how to download of software called Mega. So how to use Mega, how to make phylogenetic trees using Mega software. And you can always ask me about phylogenetic analysis of fungal for the taxonomic analysis for taxonomic purpose. I'll be happy to answer any such questions. So I would like to make these next 3 hours quite interactive. So I have switched. I can allow all of you to speak up whenever required. So I have not turned off your mics. How many of you have already used Mega software? Just raise your hand. From 81 or 80 people, only 10 participants have already used Mega softwares. So I would like to know from these 10 participants, what are your expectations from today's sessions? One of you can answer.
Hello, sir.
Yes.
Sir, I am PhD student from Punjab Agricultural University. I just want to update the things what I have already known. That's the one important thing. And I just want to compare how these Mega 11 and RAXML is differ and which software I have to use, which is the more advanced one, what type of information I can get a difference from this Mega, or I should go for a RXML. The recent papers are coming with RAXML only. So these kind of information, like additional information, I want to know from these training programs. Thank you.
Thank you, Kopuraj. Any more comment? Any more response? For the question, how many of you have already used Mega software? Yes, madam.
Good morning, everybody.
Good morning, ma'am.
I have used Mega software, but I didn't have a formal training using it, so I'm expecting to know more about Mega, how to use it, and also transfer the knowledge to others.
Yes, thank you so much for that. Any more response?
Sir, I want to add one more thing. I just want to know how to select the out groups also.
Yes, we will discuss that. We'll have almost 3 hours with us. Okay, please feel free to ask any questions to me. Keep your questions ready. In between or at the end of some segment of this session, I will be happy to answer all the questions you may have. Okay. People who have not used Mega so far, what are your expectations from this training program? May I know, please? Some of you can just speak up. I hope the question is clear.
Good morning, sir.
Yes.
Sir, actually, I'm a beginner regarding this. I really don't know anything about this, so I wish that I could able to get the basic knowledge regarding that.
Yes, Parul. Yes, I will be spending first 45 minutes explaining the concepts behind phylogenetic analysis. Okay. Then after that, we will have question and answer sessions. Of course, in between also you can ask questions. After that, we will take a break for 10 minutes. Then when we come back, we will start with the installation of Mega software in our laptop. Make sure your laptop is ready. Make sure your internet connection is good enough for the downloading Mega software. Thank you, Parul. Any more response from the participants who have not used Mega software so far?
Good morning, sir. I have used Mega several times, but later I found a problem that when I submitted the publication, the reviewers always asked for, they were reluctant to accept it, the phylogeny on the basis of Mega. That's why I switched to some other like Rexmail on Cypress portal. So for the students, I think for the beginners, it's a good software. But I am interested to know what are the differences that exist between different softwares like Mega and Rexmail, which is available on different portals. And secondly, how different models that we select affect the phylogenetic tree analysis, like neighbor joining, maximum likelihood, and others. Thank you.
Thank you, Dr. Munazar. Thank you for that. To be frank, these 3 hours or 2.5 hours will be for all this will be for the beginners. Okay. Of course, there will be some insight shared about the advanced phylogenetic analysis. Okay. I will also give information where you can find information about advanced phylogenetic analysis, because MycoAsia has already conducted one online training on advanced phylogenetic analysis. Dr. Mubashir Raza has already conducted it, and the video is available on YouTube, so our YouTube channel. But anyone who is interested to know more about phylogenetic analysis, advanced or basic, I'm sure they will be benefiting from today's session. Yeah, thank you. Any more response? Because we cannot be talking, I cannot be talking for the next 3 hours. I expect this should be very interactive and very dialogue type actually. Yes, Edwin.
Okay. Thank you, sir. Good morning.
Good morning.
The Mega software has measured to contain a large collection of the methods and tools of computational molecular evolution. Here we describe new addition that make Mega a more comprehensive tool for building time trees of species, pathogens, and gene families using rapid relaxed clock methods. Uh, yeah, I'm experienced the method for estimating divergence times and confidence interval are implemented to use probability densities for calibration, constraint for not dating, and sequence sampling. Thank you.
So we'll be able to cover some of the things what you are expecting from us today, but not all of them. But we hope that many of you would be benefited because many of you are beginners, as I understand. At least 85% people are not aware of, have not used Mega. So, but we will cover some of the aspects you have mentioned right now. Okay, thank you. And any more response, Shubham? Or anyone? Okay, so let me start with the presentation. Yes, okay. Good morning again, ladies and gentlemen who have just joined this session on phylogenetic analysis using Mega. See, today we will be spending the next 2.5 hours discussing about phylogenetic analysis of fungi, and especially using a software called Mega. As you already know me, but let me also give some information about MycoAsia journal. Some people, I mean, I have muted all of you. Coming back to about to MycoAsia journal, we started this journal in 2019, and during this course of its development, we realized that many of the authors, especially young authors, need some training in some advanced aspects of mycology. So we started giving online training to young people, young students from all over the world. I'm one of the co-founders of MycoAsia Journal, and if you have any questions about MycoAsia, you can always email us at contact@mycoasia.org, and I am also available on WhatsApp. You can email, not email, you can send a message to my WhatsApp. You can just send a message to +919763158247. Okay, so this is the logo of MycoAsia Journal. I hope many of you are familiar. And this is the logo of MycoAsia Network. So MycoAsia Network is envisioned to play an important and beneficial role in coming years to connect mycologists and mycophiles of the world. We hope to connect people from different backgrounds, different nationalities, different geographical regions, and and different educational backgrounds, educational levels, and they interact together and get benefited from this networking. So this is MycoAsia Network's recently designed logo, and we have done some around 15 events, online events so far. You can go to this website and get details about the upcoming events and also about the past events. Coming to today's topic, I'm going to talk about Mega software in the next 45 minutes or so. What is Mega? Mega is a computer software. It is not available for the mobile phones, but you can use Mega on your desktop computer, on your laptop. Okay, the full form of Mega is molecular evolutionary genetics analysis. The main functions of or main features of MEGA is that MEGA has been used to conduct statistical analysis of molecular evolution. If you have a gene sequence data or a protein sequence data, you can conduct statistical analysis for molecular evolution. Of course, it's very well known that MEGA can be used to construct phylogenetic trees. So this is the website of MEGA. Okay, let us go to MEGA's website. Romana, can you see the website?
Yes, it's visible, sir.
Okay, thank you. See, this is the website of our MEGA software. Okay, so there are certain, you can download MEGA software from this option. Here you just say download, it will be downloaded. Okay, you can, if your system is a little bit different, you can select, okay. And if you want graphical interface or your command line, you can find out if you want for Windows, Mac, or Ubuntu, or Red Hat, or other Linux. So there are many options out there. Okay, so there are some options. There are some. There is a feature called tutorial that allows you to go through some videos. Okay, for example, here is a video on molecular dating with MEGA. And also there is one video on choosing and acquiring sequences. So meant like that actually. And if you there is a video on bootstrap with MEGA. So and there are some books and articles. So these are the books. You can, but this book has to be purchased on Amazon. And this again, one more book, Phylogenetics, that the previous book's name is Molecular Evolution and Phylogenetics, written by Ney and Kumar. The second book is Phylogenetic Trees Made Easy, how to manual. So this is available on online platforms. So you can download the oldest paper on MEGA. This is 1993 paper on MEGA. There's one more paper from 2018, 13, 18, so many more, okay? So there is a walkthrough. So it helps you, in understanding MEGA software and how to use it, okay? Especially how to install MEGA. Installing MEGA, for example, here there is a clear-cut instruction how to do all these things, okay? For example, if you want to, for example, if you want to know about how to construct a likelihood trees, so just click on constructing likelihood trees. So the features of some of the features of MEGA, what MEGA can do phylogenetic analysis, it can do bootstrapping, it can do distance and diversity analysis, it can do model selection, it can do substitute pattern analysis, it can do rate variation, ancestral sequence, clock test, time tree, selection test, disease mutation. I'm not going to talk about all of this, first of all, because I am not expert in all the aspects of MEGA. I can do phylogenetic analysis, tree construction, model selection using MEGA. Okay. There is some documentation manual, online manual. You can say here is on online manual. Again, some links to publications. Same link, same publication. So you can always give feedback. Okay. So these are the original authors of MEGA software: Ney, Sudhir Kumar, Tamura, Stetcher, Peterson, and one more Peterson. This is developed by Pennsylvania State University, and this initial release was was in the year 1993. So the mega is 30 years, almost 29 years old now, actually. So when I was a PhD student, it was not very popular, though it was available. It was not very popular for the reasons that were not accepting the tree developed by Mega Software. I mean, the criticism is still there, but I don't think it's very much valid now because Mega has developed over the years, kind of transformed now actually. As I told you, this Mega is available on OS, Mac OS, Linux. It's platforms x86, x86 to 64, available in English language only, and it's a proprietary freeware. You're free to use it, but it's a property of someone else, but you're free to use it actually. I have already explained the features and other stuff about Mega. So why Mega? I mean, of course, last year in the year 2022, I have already took 4 sessions on Mega. Why I'm so so much in favor of teaching Mega? Because it's easy to use. Because when I started my PhD journey, of course, I had to construct phylogenetic analysis. So I had to do phylogenetic trees. But that time, POP was very popular. I think POP is still popular, but POP is not like very easy to use because you needed to do the scripting kind of coding kind of analysis. This scripting you have to do actually. You have to give command lines and all those things. So that was POP was not easy to understand, to use also, but Mega, in comparison, Mega is very easy now. Just click, 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 and as I told you, Mega is best suited for beginners. It's free to use, as I told you, and since 1993, I have seen a great transformation in Mega. It's now popular with new features, like I see disease related to disease. I see features on molecular dating and other stuff actually. Going towards the concept of phylogenetic analysis, let's talk about the tree of life. The diversity of life on this earth can be represented in the form of a tree. Okay, this is a phylogenetic tree of life actually. It is similar to an organic tree. So a tree is expected to have a root. Okay, then it will have a kind of a trunk and the branch and leaves. Similarly, you have the tree of life here. You see, here is the ancestral, hypothetical ancestral, and you have many branches. Here is the branch of plants. Here is the kind of a branch of mushrooms, kingdom fungi. Actually, here you have animals. Interestingly, fungi are closer to animals than to plants. That's quite interesting because fungi have been taught in the Department of Botany actually. The fungi are still, mycology is still taught in botany department, but interestingly, phylogenetically, fungi are closer to animals than to plants. Okay. So what is a phylogenetic tree? A phylogenetic tree is nothing but a graphical representation of evolutionary relationships. Okay. So I will talk about different parts of phylogenetic trees. Before that, let me give some information about the taxonomy, the science of taxonomy. What are the components of taxonomy? So taxonomy is a science. It is not just a tool. Okay, I mean it is not a tool. Taxonomy is a science because taxonomy is all about hypothesis testing. Like any science, taxonomy is also a science because when somebody says these individuals, these fungi should be clubbed together as a species, that is a hypothesis. We can test the hypothesis and prove or disprove it. So therefore, taxonomy is a science, and it's a beautiful science. We have great champions of this science of taxonomy like Carolus, Linnaeus, Charles Darwin, many more actually. Okay, taxonomy is nothing but the science of naming, describing and classifying organisms. Okay, taxonomy has 3 components. First one is identification, second one is classification, third one is nomenclature. Identification is a binary process. It's an important process. It's important aspect of taxonomy, and because important and very utilitarian. Identification is sought regularly by experts, non-experts, general public. You can always find on social media what is the name of this fungus, what is the name of this mushroom. So identification is quite in demand for that matter. And it's just, what is identification? Identification is a binary process by which we can determine whether one particular organism or a fungus belongs to a known group of organisms or fungi or not. Or in this case, whether a group of whether an organism belongs to the known group, it has to be described as a new taxon. It's a binary process. It's like your Aadhaar card. If you just give your Aadhaar card, you will be identified. So if your data is already there, you will be identified. Similarly, if your data, otherwise your data is not there, you will not be identified. It's a binary process, yes or no. That's it. Okay. Then about the classification. Classification is the arrangement of organism based on similarity. It is the core of taxonomy. Classification is the core of taxonomy because this is the hypothesis testing. Where we arrange organisms based on similarity. Similarity can be based on morphology, external appearance, whether these mushrooms are similar in we have same color, or whether they are having different colors. I mean, something similar line. Okay. If the external morphologies are not very meaningful in terms of classification, for classification purpose, then we might use DNA sequence data. Since 1990s, we have been using the DNA sequence data, especially in mycology or in the taxonomic analysis. So now we use both morphology and DNA sequence data in combination or separately in taxonomic analysis. The last one is nomenclature. This is also an equally important aspect or component of taxonomy. It's not a science because nomenclature is nothing but a set of rules. Okay, it governs naming of organisms because nomenclature governs this scientific communication, the correct communication of the taxonomic knowledge, or and these nomenclature or these rules are developed and enforced by taxonomists. So it's a mostly democratic way. Now, modern taxonomy includes phylogeny as an integral part of the classification. Now, important question is, what is phylogeny? Okay. Can somebody tell me what is phylogeny? I will switch on. I will allow you. Can you raise your hand and just let me know what is phylogeny? Yes, Tanya.
Sir, it's the evolutionary studies and identification of the organisms.
Okay. Anyone else? What is phylogeny?
So it is basically the relationship between the different families which were there during evolution.
Families. Okay.
Yes, sir. The representation of evolution and relationship.
Okay. Any more response? Thank you, Amber. Jagdishan?
Sir, it's the history. We can check where it comes from.
History of whom?
The particular organisms we're comparing with related species or with other groups.
Okay. I will come back to the concept of phylogeny again in the coming slides. Any questions about taxonomy, the different components of taxonomy, and how they are like connected and how they are very important? Any questions on those? Tanya? No?
Sir, I'm studying biochemistry. I don't know detail about the evolution and the taxonomy. Can you tell detail about the taxonomy and phylogenetic system?
I have already explained taxonomy. I hope you have focused on that.
Yes, sir. I'm focused.
Okay, now I will come to phylogenetic analysis. Therefore, I ask the question, what is meant by phylogeny? Okay.
Yes,
Purushottam.
sir. Yes, sir. Okay, sir.
Any more questions or any response? Shubham. Okay, looks like you can keep your questions ready when I come back to you after 10 minutes or so. Okay. See, coming back to phylogeny, phylogeny is based on the premise that genes can be used as a evolutionary chronometer. You understand evolution. Evolution is something might have happened or something has happened. So that's evolution. Chronometer is what is chronometer? So chronometer is something which tells about the time. Okay. So genes can be used as an evolutionary chronometer. So that is the basis of your phylogenetic analysis. So phylogeny is nothing but the evolutionary relationships of organisms and genes. Okay, the organisms can be studied at different levels. For example, species, genus, and your family. Somebody mentioned about families. Then you have orders. Then so on, phylum, blah blah blah. Similarly, you can study the evolutionary relationships of genes. Okay, so that's also part of phylogeny. So as I told you, genes can be used as evolutionary chronometers. For that matter, even proteins can be used as evolutionary chronometers. Basically, these evolutionary chronometers measure the evolutionary change. So measure in terms of time scale. Okay, here it is not 24 hours. We are talking about millions of years. Okay, and what kind of gene or markers, what kind of genes can be used as evolutionary chronometers? For example, 16S rRNA gene in case of prokaryotes. Why it is so popular? That's because these genes are present in all members of target groups of prokaryotes. And these are giving an example. Any marker which you want, which we want to be used as evolutionary chronometers, it should be present in all members of the target group. It should be functionally homologous in the organism. Okay, homologous. That means so they should have same ancestry. They should contain regions of conserved sequences for comparison. There are two kinds of regions: a conserved region and variable region. Conserved regions helps in the alignment of the sequences, and variable regions have phylogenetic signals. So you should have both conserved regions and variable regions. And the variable region should not be too variable. I mean, these variables should be these variable regions should be slow enough that we can measure the evolutionary changes. If there are some fragments in ITS region which are we cannot use them in the phylogenetic analysis, we just exclude them from the analysis. I mean, basically, in the housekeeping genes, you cannot expect rapid changes. Okay. This is a list of markers that have been used in phylogenetic analysis, especially also in identification of organisms. For example, animals, we use cytochrome oxidase. In plants, we use RBCL. In bacteria, we use 16S. In fungi, we use ITS. In protists, we use ITS. There are other markers also. This is from Wikipedia page. You can always use. Go back to Wikipedia and see for updates. So typically, this is how we do phylogenetic analysis. At least in case of fungi, we have a culture of fungus. Okay, then we probably do long-term preservation of it. For then probably from one copy you extract DNA. Then you do PCR for the amplification of desired region. In case of fungi, it could be ITS to start with. Then once you have PCR products, you can do the sequencing of the desired genes. I mean, especially ITS in case of fungi. Once you have genes sequences, this is a chromatogram here. Then you can extract gene sequence from it. You have to do phylogenetic analysis. Phylogenetic analysis will give an idea about the comparative position of that particular sequence on the tree of life, or in case of fungi, that segment of tree of life of fungi or fungal tree. Okay. And the phylogenetic tree concept of phylogenetic tree is not very recent. Like this was known to Charles Darwin. This is from Charles Darwin's notebook, where he has represented evolution of a group of organisms in the form of a tree from on his notebook. Similarly, we have three domains: bacteria, archaea, and eukarya. So these are the highest level of the domain is the highest level of taxonomic category. So now coming to parts of a phylogenetic tree. Can someone tell me what are the parts of a phylogenetic tree? Yes, can someone please raise your hand? Let me know. Anyone? Yes, Amber.
So branches are there, and then we have nodes, and the main is from where everything a branch up is a root.
You said branches. You said nodes and
So roots.
Roots also, right? Okay.
Yes, sir.
Okay. Do we always need?
Yes, sir.
Always need to have a root in case of a phylogenetic tree? Can the tree be unrooted? Any idea, Amber? No.
Well, sir, it can be unrooted. Like if we do not know the proper history of the origin, then the roots might not be there.
Yes, sure. Yeah. That's a good answer, actually. Thank you. Any more response? What are the different parts of a phylogenetic tree? Anyone wants to add here? No?
Ancestor.
Yes. Please.
Ancestor.
Ancestor is a part of the tree. Okay.
Yes.
Let's see. Okay. Any more response? Thank you, Shubham.
So phylogenetic
Taxa,
tree
lineage.
include grouping the similarities or in the sister groups, and the other ones are grouped into the outgroups that are not similar to the sister taxa.
So outgroups are a part of phylogenetic tree. You mean to say?
Yes, sir. Yes,
In-group
sir.
and outgroups are the parts of phylogenetic tree.
Yes, sir. So somehow or the other, it is linked to the ancestral part. It is
Okay.
rooted.
Okay, Ramit Kundu, what is your response? You have raised your hand. I thought you might like to respond here. Okay, no, I guess. Probably you are saying hi to me. Good. Anyway, I'll continue. I'm going to explain about the different parts of
Terminal branches are actually known as the leaves of a phylogenetic tree.
Okay, Ramit, thank you. What is that?
Yes. Terminal branches are known as the leaves of a phylogenetic tree.
Leaves. Okay. Then
that's it.
only leaves.
I think almost leaves. Yeah.
Okay, thank you. Okay, I'll switch. I'll mute you guys now. I will just continue with the presentation. Okay. Ramana, can you see the slide here?
Yes, sir, we can see the slide.
Okay. See, here you get an idea about different parts of a phylogenetic tree. Okay. First, you have a root here. That is the oldest ancestor, or at least hypothetical ancestor. Why I'm saying hypothetical ancestor? See, evolution might have happened millions of years back. So we may not have correct picture about the root, that particular ancestor now. So then we assume that that particular taxon could be the ancestor of these organisms here. These organisms mean these are the organisms. These are the present day organisms. Okay. So first part of a phylogenetic is the root. Okay. Second, then you have branches here. These are the branches. Then you have nodes. Nodes are these are represented in the form of circle here. There could be two types of nodes. Internal nodes, which are inside, inside before the leaves. Okay, these are internal nodes here, and you have terminal nodes. So internal nodes actually represent the hypothetical ancestors of these. So these are the present day organisms. These are called terminal nodes or leaves. And these represents genes, population, species. Okay. So so two nodes are connected by a branch. Okay. Here between these two branch. So branch length can vary. It need not be fixed the length. So that length will give an idea about the amount of evolution that might have occurred. So root, branches or lineages, internal nodes, and terminal nodes are the branches of a phylogenetic tree. Is this clear? Is this clear?
Yes, sir.
So we have root, we have branches, we have nodes. There are two types of nodes, internal nodes and terminal nodes. Terminal nodes are the present day taxa, or this could be genes, population, organisms. Okay, and the internal nodes are the hypothetical ancestors of these leaves or terminal taxa or terminal nodes or taxa. I hope this it is clear now. Any response?
Yes, sir.
Yes, sir.
Okay, great.
Yes, sir.
Okay, great.
Yes, sir.
Let me continue. Now the important question is how to construct a phylogenetic tree. To construct a phylogenetic tree, first you need to assemble a data set. Data set could be gene data set or protein sequence data set or morphological data set. Once you assemble a data set, I will tell how to assemble a data set. Then we need to do the multiple sequence alignment. We can use Mega to do the multiple sequence alignment. Once you have a multiple sequence alignment, we need to find out the best tree or best trees. Some phylogenetic tree construction result in one tree. Some of them can result in multiple trees. Okay. For example, maximum parsimony method can give you multiple trees. So once you have branching pattern, you need to know the statistical support for the branching. That will be done using bootstrapping. Again, finding out the best trees can be done using Mega software, and bootstrapping of the that particular branching pattern for the branching pattern can be done in Mega software. Then once you have the tree with statistical support or bootstrap support, can be presented and can be edited here and there for aesthetic purpose. So that can also be done in Mega software. So coming to the assembling of data set, you can have your own sequences. I mean, normally a researcher will generate his own sequence, sequence in the sense for in case of fungi, it could be ITL sequence, in case of bacteria, RNA gene sequence data. Okay. Then once you have your own sequences, then you can retrieve homologous sequences from public database. Very common one is NCBI. Okay. Other ones are also there. In case of bacteria, you can go to easy taxon. Now the name is changed. I think it is a little bit different now. Basically, these are the public databases from where you can download sequences or homologous sequences of your of the newly generated sequences in your lab. How can you find out the homologous sequences? There could be two basic kinds of search strategy. One is actually you can just type the name of the organism. For example, you just go to NCBI and type Aspergillus niger, then you will get related sequences. Or you can paste your sequence on NCBI server on the NCBI page, that is called BLAST. Do the BLAST, then you will get the homologous sequences. These are the two basic kinds of search strategy. So Now you have your own sequence plus homologous sequences from the public databases. Now you need to create the multiple sequence alignment. So this is a graphical representation of a unknown data set. Okay, this is how a multiple sequence alignment looks like. So these are columns. Okay, these individual, this is, I mean, this is like an Excel sheet. You have a column here, your columns here. The column can have similar bases or it can have dissimilar bases. So then when there are dissimilar bases, these becomes character states. So this is a one character. So then these are A and C are character states. Maybe it might sound a little bit complicated, but just remember each column is a character. Okay, and the column can have multiple, can have dissimilar character, dissimilar bases or nucleotides. Then this becomes character states. For example, in this column, this is the character, and character A is a character state, and C is another character state. Sometimes gap can also be considered as a character state. Okay, these are the popular multiple sequence alignment program of software. Mega, I used to use CView. You have MAFT server. If you have around 500 to 1000 sequences, the Mega may not be the best program for multiple sequence alignment. So then you can go to a web server called MAFT, which will do the job very fast and very efficiently. Some important concepts. Each column, every residue from every sequence is homologous. So this is a residue. For example, all these residues are believed to be homologous. See, homologous or homology is a very important concept. It is not like similarity. Homologous is an hypothesis. Okay, so try to read about it if you are not aware about it. Sometimes your alignment may be little bit not very good, so you can adjust here and there. Don't adjust too much. Otherwise you will be inserting bias in your analysis. We normally use DNA sequences for closely related sequences and amino acid sequences for distant relations. Like when I was doing a computer course, I was told about this phrase: garbage in, garbage out. It is easy to make a phylogenetic tree, but what kind of sequences you add to your multiple sequence alignment, that will determine whether you're going to get a garbage or a reasonably good tree. So make sure you add quality sequences in your data set, which are in terms of taxonomically, they are very good, and they are quality in terms of sequence quality, they are good actually. Now I'm going to talk about tree building methods. We have a multiple sequence alignment. Let me go back to this process, how to construct a phylogenetic tree. First is to assemble a data set. That is to have your own sequence, then retrieve homologous sequences from public databases. After that, you create a multiple sequence alignment that can be done using Mega software. Now, how to find out the best tree? Before that, I want to ask whether you have any questions. Let me just unmute all of you.
I think Govardhan Sahu is having a question, sir.
Yes, Govardhan. Yes.
Sir?
Yes.
Sir, can you explain what is bootstrapping? Because I am a beginner also. I don't have any background in molecular biology. So I just want to know what is that bootstrapping.
Thank you, Govardhan. Nice to hear from you. Thanks for joining in. I have already added a couple of slides on bootstrapping. I will be explaining about bootstrapping at the end of this presentation, almost at the end of the presentation. Please wait. Okay, I will be explaining it very clearly. Okay,
okay,
then
sir.
If you still don't understand, then I will explain it to you.
Okay, sir.
Yeah.
Okay.
Two people have raised a hand in the audience for question, Kaustav and Kupura Jagdishan. So Kaustav first.
Hello, sir.
Yes, Kaustav, yeah.
My question is, which type of file format we have to insert in your Mega software?
There are two formats which would be accepted by Mega. One is FASTA, another would be Mega format. You can always convert your FASTA into Mega format.
Okay, sir. Thank you.
It's like you take set of sequences and and you paste the sequences in FASTA format, save it as a text file, and import it. Okay.
Okay, sir.
Pratika?
Hello, sir.
Yes.
Sir, can you please explain the parts of the phylogenetic tree once again,
Yes, I will
please?
do that. I will do that.
Yes,
Okay.
thank you.
Dean? Moidin?
Hello, sir. Hello.
Moidin, tell yeah.
Hello, sir. How to do morphological data set, sir?
A morphological data set is like you have different characters. For example, mushroom color. It is color.
Sir.
For example,
Sir.
it is, for example, it's highlight. I mean, not mushroom. Forget about mushroom. Just mycelial character.
Yeah,
Okay.
yeah, sir.
Whether it's hyphae is highline, hyphae is demetaceous or non-demetaceous.
Okay, sir.
So you can have that particular character. Then you can, if it is more demetaceous, you will make it one. Otherwise, if it is otherwise, you will make it zeros. Okay. Similarly,
Okay,
you can develop morphology. It is, see, the cladistics developed from the morphological data set. The concept.
Yeah, sir. I know, sir. I know, sir. Sir.
So all this modern phylogenetic analysis has the basis in terms of morphological data sets and their cladistic analysis. So
Yes, sir.
people still use morphological data sets and they do analyze it. And you can sometimes combine morphological data with the DNA sequence data. That is also possible, but you have to wait.
Yeah, sir. Sure.
You have to weight them accordingly. Whether you want to give more weightage to morphological data, one particular data, it's all around character. All those things will matter actually. Basically, I have not combined it before, but I read a paper which explained combining different types of data. Morphological data, DNA sequence data. For example, Dr. Vinay is telling size of the conidia, all those matter. Even qualitative data, quantitative data also you can actually
Yes, I know, sir. Actually, morphological data, we don't know any genetic data. Only we boot up only morphological data, sir.
That should be possible, but then you use only very simple type of analysis, cladistics, or what you call dendrogram type.
Yeah. Okay, sir.
Okay, Pratika, you had a question?
Yes, sir. I did ask about the parts of the phylogenetic tree.
I will. Just keep your hand raised. Okay.
Okay.
Lower. Jagdishan, Kupraj.
Yes, sir. Sir, you have mentioned that during multiple sequence alignment, we can adjust the sequence. I want to know why we need to adjust the sequence and what is the importance of adjusting and when we should do the adjustment. And if we are doing more adjustment, it creates bias, you said. And can you explain that how it will create a bias and what is the importance of adjustment?
See, in public databases, you may not get sequences of same length. Okay, some may be longer, some may be smaller, right? When you align them, there will be kind of a length issue will be there. So you need to cut some of the initial portion. Similarly, at the end of the data set. I'm going to do the demonstration and I'll explain to you. Okay? Because during the demonstration, I will explain you that. Okay?
Okay, sir. Thank you, sir.
Okay, Amber.
Sir, you just talked about homology. So my question is that Darwin proposed a lot of his theory based on homology. So how much can we say is the evidence about the common structure is still prevailing in this modern era biology? Do we still have, can we still take homologous structure as a very important point when we talk about evolution, or some certain things have changed?
I think, see, the evolutionary tree is built based on the ancestry, right?
Yes, sir.
And homology is about the ancestry of the characters or the residues, right? So we need to use connect the present day nucleotides with their possible ancestors, right? So homology is a very important concept. in terms of multiple sequence alignment. And homology is, as you are definitely knowing, that it is different from similarity. So
Yes, sir. Yes,
that's
sir.
what I explained. Yeah, we will discuss about this later. But thanks for that. It's a very good, very
Okay,
good
sir.
question. Maybe some of us may not be understanding it very clearly at the moment. Okay, madam. Yes. Ikichi. You have any question? Any more? Anybody? Okay, I'll just meet all of you now. I will go back to the parts of phylogenetics. Okay, Pratika, you asked me about different parts of phylogenetic trees to be explained again. The first part is the root. Or let me start with the terminal nodes this time. So these are the present day taxon. Or in our case, these are the sequences what we use in our phylogenetic analysis. These are called leaves or terminal nodes. These could be gene sequence data or populations or species conceptually. Okay. Then present day sequence or present day taxa, this particular taxon is connected to the ancestor, hypothetical ancestor, through a branch. This is a branch, and this is the internal node, which represent the hypothetical ancestor of this taxon. Okay, so you have terminal nodes, you have branches. Okay, then you have hypothetical ancestors of these present data sequences. These are internal nodes. You can see that terminal nodes are colored in black, and the internal nodes are colored in white actually. So these are the meeting point of the taxa actually, or the sequences where the divergence might have happened actually. Then at the beginning of all this, we have the oldest ancestral taxa. That is the root. I'm going to talk about the routing, or the root at the end of this presentation or later part of the presentation. So I was talking about how to construct a phylogenetic tree. First, you need to assemble a data set. Then you need to have a multiple sequence alignment. Okay. Multiple sequence alignment we already got using Mega. Now you have to find out the best tree. Okay. Before that, we need to understand we can build phylogenetic trees using different methods. Okay. There are basically two kinds of tree building methods: distance metric methods and discrete data or character based methods. Remember, there are majorly two kinds of tree building methods: distance matrix methods and discrete data or character based methods. So neighbor joining and UPMGA belong to distance matrix. They are generated using distance matrix method. Okay. They belong to they belong to distance matrix method. So neighbor joining trees, UPMGA trees, they belong to the distance matrix based trees actually. Okay. Then we have parsimony trees or parsimony method, maximum likelihood method, Bayesian method. They come under discrete data method actually. So parsimony trees, maximum likelihood trees, Bayesian trees are generated or constructed based on discrete data method. So what is the difference between distance matrix method and discrete data methods? Anyone has any idea? Let me unmute people. What is the difference between discrete data method? Anyone? What is the difference between distance matrix methods and discrete data? Anybody? Anyone has any idea? No? You can speak up. The microphones are unmuted actually. Anyone has any idea? What is the difference between these? Any hint? Any hint? Any guess? No?
Maybe bootstrap, sir.
No,
Values
nothing to do with bootstrap, Jagdish, Kupuraj. No. Any guess? People who have already used Mega, what do you think about this?
Non-distance.
Oh, nothing to do with the long distance or short distance, Shubham.
Not
Sir,
long.
Sir, from place to place, different place to different place, the organism is classified and then character based. Whether the snake is venom or not, based upon the characters or day-to-day life activities, we can classify the organism.
Thanks for that, Purushottam. Okay, any more response? Okay, thanks for this. Very nice that you have at least you guys had the courage to respond. Very good. Okay. Basically, this is a very important concept you should understand about distance metric methods and discrete data methods or character-based methods. Let us talk about distance metric methods, which include neighbor joining and UPMGA majorly. Here, the multiple sequence alignment is first converted into a data matrix. So that data matrix is used as used to construct the phylogenetic tree. So that's the specialty of data matrix methods. So where the multiple sequence alignment is first converted into a data matrix, that particular data matrix is used to construct the phylogenetic tree. In case of character-based methods or discrete data methods, which include parsimony, maximum likelihood, Bayesian methods, multiple sequence alignment is not converted to a data matrix. Instead, the alignment file is used as it is to use used to construct the phylogenetic tree. Okay, I'm explaining again. In case of distance matrix methods, multiple sequence alignment is converted into a data matrix initially. That data matrix is used to construct the phylogenetic tree. In case of discrete data or character-based method, multiple sequence alignment is directly used as it is for the tree construct. I think somebody has some question. Purushottam, wait, Purushottam. Yes, Purushottam, yes.
Sir, how the sequence is converted to database? Sir?
That is done by the program itself.
Okay.
We don't have to do any efforts on it. You have to just click. Okay, when you say I want a neighbor joining tree, the Mega will say you have given me the multiple sequence alignment. I will first convert into a data matrix. I will use the data matrix for the tree construction. You will get the tree in 5 minutes. Deepak.
Okay, sir.
Sir, good afternoon. Sorry,
Yes.
good morning, sir.
Good morning.
Sir, discrete matrix or discrete data, which is the better for the construction of tree?
Good question. We'll come to that later. Thank you. Yeah.
Okay, thank you, sir.
Prabhu. Oh, okay. Any questions? This is an important concept. Hope you have understood. Hello?
Yeah, sir, understood.
Yes, sir.
Please go ahead.
Yeah, thank you so much. Yeah. I'm explaining again, in distance matrix methods, multiple sequence alignment is first converted into data matrix. That data matrix is used by the program to construct the phylogenetic tree. So neighbor joining and UPMGA belong to the distance matrix method. In case of discrete data or character-based methods, multiple sequence alignment is used as it is to construct the phylogenetic tree. The data matrix part is not there in the character-based method. Parsimony, maximum likelihood, Bayesian tree methods are the examples for discrete data methods. Now, important question is about, see, you got a tree. Okay, for example, here you got branching pattern. In science, statistical support is very important. You should not leave anything to chance. In the sense, even if you leave it to chance, you have to quantify the probability of that particular branching pattern because of a chance. So that is how we are into bootstrapping. We use bootstrapping to tell whether this branching pattern is statistically supported or not. So what does the Mega software do, or any bootstrapping software do actually, or phylogenetic analysis software do? First, it uses a multiple sequence alignment, and the software takes random subsamples and build trees from subsamples. Then, for example, if we ask the software to take 500 subsamples, it will take 500 subsamples, and based on for each subsample, a tree will be generated, and the tree will be at the end of this process. All these subsample trees will be combined. Then the frequency of occurrence of different branches of different lineages will be represented in percentage. This is a bootstrap value, 100%. That means if we said Mr. Mega, you do 100 mega subsamples, out of 100 subsamples, all the samples showed this kind of branching pattern. So that means this kind of branching pattern, this branching pattern is statistically well supported actually. This is when bootstrapping is to test the phylogenetic accuracy of the tree. Normally, bootstrap values higher than 70 likely to indicate reliable groupings. Okay, if the bootstrap support is less than 50, then there is a likelihood that the branch will collapse, symbolically, that means that kind of branching pattern is not statistically well supported actually. There is a YouTube channel, YouTube video. You can watch it. It's like 10 minutes or something. I'm not going to spend 10 minutes. This YouTube video will give a good idea about bootstrapping and the importance of bootstrapping actually. Any questions about the bootstrapping? Sahu, Govardhan Sahu.
So now I think it is clear. Thank you, sir.
Thank you, Sahu. Yeah.
Sir.
Yes, Purushottam.
Sir, this method is used to identify the similarity between the one species and another species. How many genes are same between the one species or two species, sir?
No, we are talking about bootstrapping here, right?
Yes, sir. But I want to know which genes are not similar to from one species to another species. Can we find this sequence from this method?
We will discuss this at the end of the course. Sure.
Okay, sir.
Because I will be coming up with a demonstration, and you can also do the phylogenetic tree construction if your laptop is ready. Okay, and the Mega software would be installed. Okay.
Okay.
Any question, Deepak? Yes. Mr. Deepak, you have any question?
No, sir.
Okay, Madara, please.
Sir, I want to know about what is the theory behind the bootstrapping methods? What's the theory behind?
You have to read the paper by Felsenstein, 1985. Okay, basically you have to do the pseudo sampling, and you make sure the occurrence of branches in 100 replicates or 500 replicates. Okay, if some branching is because of chance, then the bootstrap support will be less. That's how the overall idea is. But if you want to understand the statistical structure or statistical design behind it, I'm not very competent about it. You have to read the paper by Felsenstein, 1985. Okay.
Okay, sir. Thank you, sir.
Rajesh Kumar. Rajesh, you have any question? You have raised your hand. Tanya.
So what does it mean that the branches will collapse?
Oh, just a hypothetical thing, just symbolic thing. Basically, it means that kind of branching pattern will not is not statistically supported actually, or is poorly supported. That's it. I mean, see, this is just you know these events have happened hundreds of millions of years. This is no branches anywhere collapsing there actually. It's just a symbolic way of telling that such kind of branchings are not very reliable or should not be taken very seriously.
Dam?
Yes, yes.
Am I audible?
Yes.
My question is like if a genus is falling within the familiar circumscription and we are not getting adequate bootstrap support, how we will justify it?
If you are relying just based on DNA sequence data, okay,
Yes.
then I would you cannot justify it. If it is in the scheme of phylogenetic classification,
yes,
one needs to have adequate or sufficient bootstrap support for the branching. Otherwise, one should not get into describing it as a new taxon. That is my personal opinion. But
okay.
how to improve that? How to the chances of getting better bootstrap support? You can use longer data. You can use longer data set in the sense you can use more gene regions.
Okay.
And you can, of course, you must be knowing this. You can use more sequences statistic in the phylogenetic analysis. But your basic question is, can we describe a new genus where the branching is not statistical supported and we are using only gene sequence data here? One should not get into this kind of adventure.
Okay. Nice. Thank you.
Thank you. Yeah. Any more question? Okay, I'll just continue with last few slides. Let me unmute you for, sorry about that. So now you have a tree. Now you have a phylogenetic tree which has got bootstrap values. Nice. So some basic understanding. Here are the branch lengths. Okay, branch lengths gives an idea about the amount of evolution estimated to occur. So you have a scale bar here. Every phylogenetic tree, I mean, most of the phylogenetic trees will have this, scale bar here. If you don't show the scale bar, this tree has almost no meaning because the scale bar tells about the amount of evolution actually. And normally bootstrap values are displayed as percentages. You can see here 100, 98, 71, like that. Some places you don't see any values. That's because bootstrap values less than 50 are not reported. Not reported means not shown in the publications because they don't make much sense actually. You can choose to root the tree. So this is the rooting. Okay. You can also you can present the tree unrooted also. What is the difference? When you root a tree, that will give an indication about the direction of evolution. What is the meaning of what is meant by direction of evolution? When we say direction of evolution, you can say which is the oldest taxon or branch here, then which is the newest one. That can be understood only when you root the tree. So any question about rooting of the tree? I think there was one question about rooting of the tree. Any question about the rooting of the tree? I think it was anyway, no question. The basic funda here is that tree can be rooted or unrooted. Okay, it's up to you. But normally reviewers will ask you to root the tree. So when you root the tree, it basically indicate the direction of the evolution actually, and how to choose an outgroup. So outgroup becomes the root actually. How to choose an outgroup? It's very simple. If you are studying Aspergillus, then you cannot. Overall, you are studying the genus Aspergillus. Then you take a sequence from the different genus. which is closer to aspergillus. So that becomes a root. Or if you are just focusing on Coltotrichum Siamens, you are studying an extensive study using 500 sequences or 1000 sequences, then you can use Coltotrichum gliospheridis as the outgroup and the root, and root it using that actually. So it's basically your study taxa, you are focusing on the in-group taxa or in-group, then the things you are not very much focused, that becomes outgroup. Okay, in case of Gunjan's study from our PhD thesis, Coltotrichum Siamens was our in-group, and in our Siamens paper, then the gliospheridis can become the outgroup, and that can be used as the root. I'm not sure how much clear it is to you, but if you have any doubt, you can always ask me later. So these are two important papers: Phylogeny for the Faint of Heart, the tutorial. It's freely available, I guess. Otherwise, you can always ask us. We will give a copy to you. And there is a Nature review paper, not Nature, I think one of the Nature journals, Molecular Phylogenetics, Principles and Practice. It's a very high advanced, it has got advanced information. Once you read this, you can go to this paper. And there is a paper by on building phylogenetic trees from molecular data with MEGA. It's kind of an outdated paper, but you can always refer to get the overall idea of phylogenetic tree construction using MEGA. We have got MycoAsia, YouTube channel. This is the link to the channel. You can always subscribe to it. We have got some interesting, videos on phylogenetic analysis. For example, how to download and install MEGA software. Okay, how to find the best DNA model for example data set, and how to construct a maximum likelihood tree, using an example data set. These videos are available on our MycoAsia YouTube channel. Anyway, I will be demonstrating about it after 15-20 minutes or so. I done this kind of session was also done, actually conducted last year. So one of the videos is available on YouTube. You can go back to it. Anyway, the recording of this today's session will be also shared on YouTube. You can come back to our YouTube channel and just access it. Our associate editor, Dr. Mubashir Raza, conducted a very useful online training on advanced phylogenetic analysis. So this is 3.5 years of, not 2.5, 3 hours of advanced phylogenetic analysis. So you can always you can refer to this YouTube video, and you can learn about phylogenetic programs, phylogenetic analysis using GAR, I think MEGA, using POP, I think then Mr. Bayes and all those stuff. Now we will proceed towards practicals. Before that, I will be happy to answer your questions. Give me a second to unmute you all. Yes, any questions now? Any questions? Please raise your hand and I'll let you ask the question. No questions? Okay, Amber, yeah.
So, when we talk about phylogenetic tree, what is the reliability that the tree which we have constructed is somewhere down the line correct with what we are trying to seek? We might have a lot of error on the way.
Okay, thanks for that question. That's a very interesting question. I mean, I read in one of the papers that phylogenetic tree is an educated guess. Okay, so it's an educated guess. That means some kind of thought, some kind of reasonable thinking has gone into it. So we can stick to this kind of premise and just accept whatever. Interpret the trees afterwards. What do you think, Amber?
So similarly, my next question is that if it's a guess, can we just further our studies with that guess? I mean, is it not very time-taking process if the initial process is wrong? Like if the tree which we have made is not correct enough, it might take a lot of time and dedication, which ultimately will not result into anything.
See, we are talking about evolutionary relationships. We are talking about evolutionary events that might have happened, millions of years back. Okay, maybe in case of viruses, maybe not so many millions, maybe recent. Even if you ask me what is the history of your family, I cannot go back to more than 100 years if it is not well documented. So here we are talking about the poorly documented evolutionary histories of so many organisms. So best bet would be to go with the assumption that the genes and proteins acts as the evolutionary chronometers, and we use genes regions which are phylogenetically rich, and then we construct a phylogenetic tree to represent the possible evolutionary relationship to the best of possible way to us actually, and we use statistical methods to just to caution people that we might be wrong. Sometimes we might be definitely wrong. So I would say you can always take a phylogenetic with a pinch of salt, so to speak. But as we don't have any other alternates, any reliable, not viable alternatives, probably we could still have some faith in it actually, till we develop something more credible and reliable. If you are quite apprehensive about it.
Okay, sir. Thank you so much.
Hi, thank you. So, Sahu.
Sir, horizontal gene transfer or maybe gene exchange actually it occurs between bacterial taxa. So, do you think that that is actually that can affect the evolutionary tree whenever we are drawing a tree based on molecular gene sequence data?
That's a problem. That's a problem in case of bacteria. So bacteriologists actually take care of that issue when they do phylogenetic analysis. Not that a serious problem in case of fungi.
Okay. Yeah. Thank you, sir.
It's an issue. I mean, I think when my postdoc mentor tried to, he was considering cytochrome oxidase as one of the as the barcode region for fungi. He was talking about paraphilia of this gene and multiple copies of it and all those things. So probably in fungal kingdom also that the issue is there probably.
Okay, sir. In fungi, okay, sir. In fungi, that horizontal gene transfer does it occur?
I mean, it occurs, but not that rapidly, that rampantly, as it happens in bacteria.
Bacteria. Okay.
And we use housekeeping genes, like ITS. ITS, there's a less chance of such things, hopefully. Mostly we have dealt with, I mean, my group has dealt with, I know, mostly housekeeping genes.
Okay. Yeah. Okay. Thank you, sir.
Thank you. Any more questions? Yes. Any more question? Okay, we'll take a break for 15 minutes. When we come back, please download Mega software. What you have to do? You have to go to. Can you see my Google, Ramana?
No, not yet. We can only see a video.
Wait. Okay. Now you can see, right? Can you see my tab?
Yes, sir. Now we can see.
Go to your Google.
Sir. Yeah.
Go to your Google and type Mega software, not Meha, Mega software. Then you see this Mega software .net. Go here. And it automatically understands your system settings and suggests these things. Just say download. Then you accept their terms and conditions. If you want to read, spend some time reading it about it. So you have to give some basic information. I'm from India. I'm a researcher. I am not from Gurgaon. I'm from CSI, NIO. This is a research institute. And I say download. See, it's around 96, 90 MB. So depending upon your internet speed, you might take 5 to 10 minutes. So when we come back from the break, hopefully your installation would be fine. I accept the agreement, you say, then you just click on next, then you next. Yes. How long is the ITS region? Around 540, madam. Yes. The folder would like to install the folder. Yes. I think you couldn't see the installation, right?
Yes, your screen was frozen in the Google itself.
So let me repeat it. Okay, just Mega software. Accept. Ramana, can you see my Google my?
Yes, sir. It's visible now.
Okay, so I'm from NIO. I'm a researcher. I'm from a research institute. Then I'm saying download. It downloads. It's around 90 MB. Ramana, can you see my executive file?
No, sir. That
At the bottom, can you see my mouse?
Yes, yes, we can see it.
Now we can see, sir.
Okay. Do you want to allow this app? Okay, yes. I accept the agreement. If you want to read it. Next. This already exists in my system. Would you like to install? Yes. Next. Create a desktop shortcut. Create a quick launch shortcut. Okay, fine. Okay, this is your Mega software. Tell me how many of you are seeing it for the first time? Okay, let me unmute. Okay, how many of you are seeing it for the first time?
Sir, I am sir.
Okay. So you have some buttons here: align, data, models, distance, diversity, phylogeny, user tree, ancestor, selection, rates, clocks, disease. Don't worry about so many things. Just focus on align, data, models, and phylogeny. 1, 2, 3, 6. 1, 2, 3, 6. Okay. Any questions? How many of you could download it?
Sir, download and install it, sir.
Okay, good, great. I think let me also share the this one link from where you can download. So let me first show you how to construct a phylogenetic tree using an example file provided by Mega team. Okay. Are you ready? Please raise your hand. Are you ready? Please raise your hand. Till I get 91 hands, I am not going to start. Okay, 44, not bad, 50%. I just passed the test, I guess. Anyway, let's start with the demonstration now. I'll just demonstrate. If you have any question, just ask me after I start demonstrating it. Okay. And I have unmuted all of you. I have also allowed you to turn on your video. Without making much noise, you can do that. Unmute yourself or turn on your video. Okay. Yes, Blaze Babu, what's the question? Please lower your hands, please. Yeah, thank you. If you go to this, let me start from the Mega, software. So here is your desktop icon. Okay, click on that. Maximize it. Do you see this button, examples? Please raise your hand if you can see it. Okay, please lower your hand. So here you have example folders. I'm going to open one of the, Mega files. Probably that would be command underscore. That would be command underscore statements dot meg. Okay, I'll click on that and then open it. Now the data set is open. It's an example data set. And you can Always color it. Color the cells. I have opened this example data set. Simply, I'll just go to phylogeny. Then I say construct neighbor joining tree. Click on that. It asks me, would you like to use the currently active data command underscore statements.meg? I would say yes. Then here is a dialog box asking for some information. Test of phylogeny, we were discussing about bootstrapping. So you just select bootstrap. There is one more interior branch test. Ignore that. Just select bootstrap. Method is already selected by the system, by the software. Number of replicates, let us keep it to 100. And let us accept all the default settings. Okay, then you say okay. Your tree is generated. So this is an unrooted tree. Okay. I'll take one of the sequences, Paris one, click on the branch and right click, then root tree. So now the tree is rooted. These are the bootstrap values, 198 and all. Now I say the tree, save current session. It saves as command underscore statements.mstx. I'm saving somewhere on the desktop. If I want to copy this tree onto the
Sir, if I can interrupt you, there's a request from the audience to repeat the procedure, please. So if you can.
It can be waited, I guess. Wait.
Okay.
So if you want to copy this tree file, tree to your Word file, then copy to clipboard, then open your Word file. Just say paste. I think that would be right click and paste. Here is your tree. Okay. You can, in the next page, you can add, or in the next page, or let me reduce it. There is some problem. Let me add the tree description. Where do I get the tree description? I go to view, not view, I guess. Sorry, caption. I go to caption, display caption in new window. Then you say copy. Then go to your Word file. You know all this tree description is copied. It says the evolutionary history was inferred using the neighbor joining method. Number one is the reference here. This is the reference for the neighbor joining. The optimal tree is shown. This is the optimal tree. The percentage of replicate trees in which associate taxa clustered together in bootstrap test, 100 replicates, are shown next to the branches. I think it's talking about these bootstrap values. The tree is drawn to scale, and here is the scale. The tree is drawn to scale with the branch lengths in the same units as those of the evolutionary distances used to infer the phylogenetic tree. Evolutionary distance were computed using maximum composite likelihood method. I think it's talking about the model of evolution. Or in the units of number of base substitution per site. Analysis involved 13 sequences. Yes, there are 30 names here. All ambiguous positions were removed from each sequence pairwise deletion option. There were a total of 421 position in the final data set that we will see. Evolutionary analysis were conducted in Mega 11. So this is the reference to Mega 11. So it was talking about 421 positions. Let me show you 421 positions. Not able to see the 421 positions. I don't know why. Anyway, it's showing only 419. Anyway, we'll fix that later when we take make our own, tree. Now the time for questions, or you can save this as this file as a file, save as desktop, my paper, my tree for paper. So now time to clarify some doubts. Yes, please. Ganga. Pratika, you have any doubt? Bhageshri? Rajnish?
So some participants are requesting it to repeat it once again.
Yeah, we will repeat it. If they have any questions, they can ask now.
Okay, sir.
Any questions about it? Please let me know whether you understood. Raise your hand if you understood. So 13 people have 16. So all of you understood, huh? No, only 13, 14.
So I have a question.
Yes, Sheetal.
Just a second. Yes. Actually, the echo is there, so I don't know why it is good. Can you hear me clearly?
Tell me, Sheetal, ask. Sheetal? Sheetal, can you hear me? Okay, any questions from anyone? Yes, Bhageshri. Ramana, can you hear me?
Yes, sir, I can hear you.
Yes, go Gordon.
No, sir.
Please lower your hands, please. Okay, thank you. Now, participants who have not understood this process of tree making that I demonstrated, please raise your hand. Okay, please lower your hands. Okay, I'm going to demonstrate it again. Just have a look. Huh? Ramana, can you see my desktop?
Yes, sir, it's visible.
Let's have a conversation about tree making. Okay, Ramana?
Okay, sir. Okay.
So, Ramana, here is a desktop here, or is my desktop, and you can see a desktop icon, Mega 11. I'm going to click on that now. Okay, Ramana?
Okay, sir. Okay.
And after that, I will go to examples. Can you see examples, Ramana?
Yeah. No, sir, not yet.
Do you
Yes.
see? Do you see
Yes.
my mouse pointer?
Yes, sir. We can see.
Okay, so I'm going to click on that.
Okay.
Examples. Then I get some options. I'm going to select a file name command underscore statements dot mega MEG. It's an example file provided by the Mega team.
Okay, sir.
I'm going to open it.
Yes.
So now the data set is open and active.
Okay, sir.
If you want to color the cells, you can color.
Yes.
Otherwise, I have colored the cells. Okay.
Yes.
So want to see how many positions are there. I don't know why it is showing 419 and out of so. Anyway, so the data set is active. What I have to do is simply click on this phylogeny.
Okay.
1, 3, 4, 5, 6, sixth button here.
Okay, sir.
Phylogeny. Then I select second option, construct or test neighbor joining tree. Then it is asking me, would you like to use the currently active data? I would say that will be command underscore statements dot meg. I'm going to select that and just accept the default settings. Then you say okay.
Okay.
And here is your tree. You want to root it, you select one of the sequences. In this case, I would like to select the last sequence. Click on the branch. You can see the green rectangle box on the branch, right?
Yes. Yes, sir. Yes, sir. It's visible.
And then I'll say I will right click. Okay, then you get a then options. Then you say root tree. So I'm rooting I have rooted the tree. Now I can save the tree session. Save current session. I will give my tree session. Select. I will save it on desktop. Okay, the session is saved. If you want to copy this tree onto your Word file, just go to Image, copy to clipboard, then open your Word file. Okay, the tree is copied here. And you want the tree description, that you'll get caption. Okay, go to caption, last button option here. Display caption in new window. Then this is for copy all text to the system clipboard. So you copy it.
Okay.
And paste it, Control V. This is how you get the tree and the description. You save it as like my tree set to second tree for paper. That's it. Now, who wants to do this? Who wants to demonstrate this? Raise your hand.
Reshma Yadav wants to do this. I think so.
What is it now? Kopura, any questions?
No, sir. I just raised for demonstration.
Any volunteer? How many of you have understood this now? Raise your hand, please. I'm going to select one of you and I'll ask you to demonstrate now. Is that okay? Lower your hands, please. Anybody, any volunteers who have understood this? It would be good if they can demonstrate. I will allow you to share screen and do the thing. Anyone, please?
Sir, I am ready.
Okay, I will allow you. Wait, let me stop presenting. I will give share screening rights. Okay, Ubraj, start. Share your screen. Ubraj, right?
Yes, sir. Yes, sir.
Are you doing it?
Yes, sir. Is my screen is visible, sir?
You have to share the present. Okay, now you are sharing your screen now. Okay.
Yes, sir.
Okay, it's visible now. Go to your desktop first. Close all the things now.
Sir, first we have to open the Mega software, and I'm in the interface of the Mega software. And here they have given the example folder. We have to click into the example folder, and it will show a lot of example folders here. I'm going to show example with the command underscore statements. It's a Mega file. I'm going to open that. Now it's showing my file is open now, and I'm going to click into that file. It is showing my sequence. It's showing my sequence, and it is not highlighted here. If I want to highlight, I just want to go to the display, and I want to go to the color cells, and it will make color. And here it's a column wise. We can see the columns here, and let's say in the first column, these all are considered as a column, and if it is something that differ from that, then it is as a characteristics. Sometimes gap is also called it as a characteristics. And
wait. Kupuraj, right?
Yes, sir.
Each column is a character.
Yes, sir.
And the dissimilar bases are character states.
Yes, sir.
Character states, not characteristics. Okay.
Okay, sir. Characteristics.
Character states.
States.
Whole column is a character. Okay.
Yes, sir.
And
Okay,
these
sir.
similar character bases are character states. In this
Okay, sir.
column, I think there are some gaps. These are also character states.
Yes, sir.
Okay. Can you please go ahead and make a tree?
Yes, sir. Yes, sir. Then we just want to go to the menu. Phylogeny here it is having. We have to open that, and there are so many methods to do the phylogeny. We are going to perform with the neighbor joining method. And here it is asking a dialog box, which I should proceed with next. So I'm going to give S, and here it's giving some parameters. Here I'm already having a test phylogeny with the bootstrap method. And if you want to change the bootstrap value, we can change. Now we are going to perform with 100 replications. So remaining things I kept is a default. Then I'm clicking next, OK, and it will finally gives my phylogenetic tree. So this is my tree. And if you here it's not showing any roots. So if you want to make a roots, I'm just clicking links from here. So then click right click, you can find the menu root tree, and it will gives root. So but in my version, I don't know, it's not showing separate scale here, but the values are changing here. Hello.
Yeah, I'm not sure why.
Okay, sir. And we have constructed this. Yeah, we can save this file, and you can go for save current session. And if I want to save in any disk, I'm just going for a desktop. Here I'm going to save my tree as a tree. and I'm just saving. It is already saved now. And to get a description for this, I am just going for a caption, and here you can see the display the caption in new window. You can get the description for that file, and here it gives the information how it is calculated, or how many nucleotides, and some with references also. And just copy these content using clipboard or a control C for copying, and you can copy this file also. Otherwise you can use the saved file and copy these all these thing in a Word file. That's it, sir.
No, first you copy the image to a Word file, Kukuraj.
Okay, sir.
How are you going to copy the, No, go to image, copy to clipboard.
So likewise, I will copy my so that's it, sir.
Save it. Thank you, Kupraj. Please stop sharing. Thank you.
Yes, sir.
Very good. Gobinda, what is your query?
Actually, sir, please can you show me from the raw data sequencing means actually after the sequence we need to do a lot of things before Mega using the Mega software because we get the forward sequence and the reverse sequence. So we need to make the conjugate sequence. After that, we use the Mega software. Am I right?
Do you have the raw sequences with you?
No, sir, I don't now. In my lab, I have, but now I don't have.
Oh,
So,
I think
but
no.
but sir, I have problem. I face one problem. When I run the forward or reverse sequence in different way, so they showing different phylogenetic because
I think see, Govinda, you see there are many beginners here. They are just some of them are really enjoying their first dream making experience. Okay, after this, once they are happy about their first experience, I'm going to take it to the little more complicated stuff. I'm going to take one sequence from GenBank, then build a data set, then create a multiple sequence alignment, then make a tree, simple tree. Then if you want to change the parameters, if you want to change the model of selection, that also we can do. But that forward and reverse sequencing, that I will not be able to do today. But definitely I'll be happy to resolve your issue.
yeah, understand, Professor. Actually, I am using the MycoAsia software for the concept sequence, making the concept sequence. After that, I use the Mega software. I think is it right or not? I don't know.
We will discuss the specific case later, Govind. Okay, thank you.
Okay. Thank you.
Now, anybody wants to repeat what we have the Kupura has done? And someone wanted to be repeated. Monica Sharma, are you here?
Yes, sir.
Do you have a laptop?
Yes, sir.
Can you share your screen? And have you downloaded the Mega software?
Yes, sir, but it's not connected. I am on my mobile and laptop. I am just checking out that way.
Okay, it's okay, Monica. Thank you. We will ask someone else to do. Madara, would you
Yes,
like
sir.
to
Thank you,
demonstrate?
sir.
No problem, Monica. Madara. Okay, tell me how many of you understood it now. Please raise your hand. Tanya Joshi, do you have a laptop with you?
Sorry, sir, I would not be able to share it.
Okay.
Sir, but I've got it, everything.
Who can share? Anyone? Please lower your hand. Thank you. Anyone wants to see the repeat demonstration? Please raise your hand if you are interested to see the repeats. Sahu, you want me to repeat the demonstration?
Sir, once more it will be better. Because
we have
I'm also a beginner. Sorry, sir. Yeah.
No problem. No.
Yeah. Okay. Thank you. Thank you.
Sahu, you have your laptop?
Yes.
And is your internet connection okay?
Not okay. It is fluctuating.
But see, you try share the screen.
Okay.
I will help you.
Okay.
You are in Andaman, right?
No, sir. Pondicherry.
Pondicherry.
No, sir. Actually, the internet connection is very poor here.
Okay, fine. Any volunteer, anybody wants to demonstrate, please? Anyone? Okay, fine. Let me repeat it for the last time. So can everybody see my desktop? Please answer.
Yes, sir.
Good.
Yes, sir.
Good. Okay, fine. I'm opening the Mega software. I'm going to go to the example folder. Can everyone see the example folder? Examples folder? Please.
Yes, sir.
Yes, sir.
Good. I'm going to select this file, commands underscore statements.meg. It's an MEG file provided with the Mega team. Okay. I'll open it. I'm not going to do anything. Just opening it. I'm directly going to Phylogenetic. Then I'm going to select the second option, construct or test neighbor joining tree. It's asking me, would you like to use the currently active data? I would say yes. And I'm going to accept all the suggestions here in this dialog box. I'm going to say OK. So here is a tree. I want to root it. I'm going to select the last sequence. I'm going to click on the branch. There is a blue rectangle box on it now. I'm going to right click and I'm going to say select root tree. So here is my rooted tree. After this, I'm going to save this tree session. I'm going to go to file, save tree session. I'm going to say my final tree session. I'm going to say that, okay. And I'm going to copy the image, I'll go to copy. I'm going to go to the third option, image. Then I select copy to clipboard. And I'm going to open my Word file. And Control V, that will be paste. And I need to copy the tree description. I'll go to caption, display caption in new window. I'm going to select all these by selecting the third option here, 1, 2, 3. After the printer, copy all the text to the system clipboard. Okay. Then I'll go back to my Word file. I'm going to say Control V. Then I will say save this file, desktop, my final tree. Thank you. Okay, so it's saved on the desktop. I'm gonna close everything. It's got results. If you want to save, you can save it. Here is my file, my final tree. Thank you. Fine. Just tell me how many of you have understood it now completely. Raise your hand. Fine. 26, 27. I mean, these are the active people. Okay. Thank you. Now, I'm going to show, please lower your hands. Thank you. Now I'm going to show you how to take a sequence from GenBank, NCBI GenBank, and make a tree. Okay, simple. First, I will go to NCBI. NCBI. Mr. Kupuraj, be ready. You're going to demonstrate again, huh?
Okay, sir.
So, have you done this before?
Yes, sir.
Okay, first, we'll go to NCBI, Kupuraj, okay?
Yes,
And
sir.
I will go to NCBI website. And I'll go to, then I select nucleotide here. And I will say Aspergillus niger IPS. And search. It is suggesting this. Okay. Aspergillus niger ATCC sequence from the type strain ATCC 16888 ITS region from the type material. Here it says blast. Just click on blast. Here, just select on sequence from type material. I want to do a proper taxonomic analysis. Click on sequences from type material. That's it. And somewhat similar sequences. And this say blast. Okay. I'm going to unselect all. I'm going to just select 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9. I think this is our sequence that we blasted. Okay. Aspergillus niger ATCC 16888 ITS region from the type material. Now what you do? Go to download. So select the FASTA aligned sequences. Here the txt file is open. Select all these, Control A, Control C. Okay, then you go to your mega. Then you say align, edit or build alignment, create a new alignment. Then you say okay. Are you building a DNA? Yes, DNA. Then you say edit, paste. Last one is just an unwanted thing, just delete it, edit, delete. Okay. Then you select all, Control A, and you go to align DNA. So DNA is aligned. Okay. You can just simply go to phylogeny, select the second one, construct a test or construct or test neighbor joining tree. It's asking me to save it. Let us do it differently. Go to data, phylogenetic analysis, protein coding nucleotide data. No, it's an it's ITS. Phylogeny, second one. How do you like to use the currently active data? Yes. I'm going to accept all the default settings. So this is my tree. I mean, doesn't look like a great tree, but our sequence is here. So what I'm going to do, I'm going to take one of these sequences. If I'm going to take whole branch and make it, out group. Click. So this is kind of one of the rubbish trees, but we have got bootstrap support of 99% here. We'll save current session. My aspergillus tree. Okay, then I copy this image to clipboard, then I will go to Word. Oops, very ugly. Caption, display caption, and then copy this. I'm going to open my Word file, not Word file, Word file. Save as desktop. My aspergillus, not dirty, it's ugly. Aspergillus tree. Okay. Just raise your hand if you have understood. Please lower your hands. Please raise your hand if you have not understood. Okay, Kupraj, are you ready?
So up to alignment and edit, I got in between step I
Okay, I will
couldn't
guide
recall.
you. Just start presenting. Yes.
Okay, sir, sure.
Please lower your hands if you are still raise your hand. Thank you.
So can I start?
Yes, please.
Yes, sir. Now we just want to go into the NCBI database, and here I'm using the organism name Aspergillus niger.
Okay, Kupuraj, please
Yes,
go
sir.
slowly.
Okay, sir.
That's what I have been told now.
Now we are in NCBI database. Here we have a search box. In the search box, we can use any organism name. We are going to take an example Aspergillus niger and ITS region. And here we have so many tools. I'm going to select the nucleotide search. So it's showing number of results, and we are going to take this first assertion, and here it's showing with the blast tool. So it will go blast with these particular assertion. It will take into the new interface blast page, and it is a nucleotide blast, so it means here N, and this is our example, the assertion number for Aspergillus niger ITS. And here we are going to limit with the type material, and we are not going for a highly similar one. Somewhat similar we are going to select, and I'm going to give this result in a new window, and I'm giving a blast. So it has shown number of results here, hit results for our query ID, means our Aspergillus niger, and I'm going to unselect all the things, and I'm just going to select how many samples we are going to prepare our phylogenetic tree. Here I'm going to take 10. I have selected 10. It is shown here, it is selected 10. And I'm going here, the menu download, and here the format I'm going to download is a FASTA aligned sequence. And it start download and it's completed. And these are all my downloaded aligned sequence for, so FASTA sequence for all the 10 selected one. I'm going to select all, and I'm copying. Now I'm coming for Meha software, and here I have to go with align menu, and I want to create a edit or build alignment. I'm going to build alignment, and here I'm going to create a new alignment. So click that and give a OK. And my sequence is a nucleotide sequence, it's not a protein sequence, so I'm going to select with the DNA sequence. And here I'm just giving a paste here. It will paste all my sequence, and you can see here the last sequence is unrelated one and it's not here. I'm just going to delete that using the right click button. And now we got our 10 sequence in the Meha software, and simply I'm going here for a phylogeny, and I'm going to construct as usual neighbor. So I think we have to save file. Sir, actually here only I forget how to save that and how to bring that to phylogeny.
That's okay. Now you will select all the sequences.
Okay, so it's selected.
Can you do the muscle alignment? After W, there is a muscle is there now. Align
Yes,
DNA.
sir. Yes, sir. Align DNA.
Just see properly whether it is properly aligned or not.
It's aligned, sir.
Just drag it from the first. There must be scrolling bar now at the bottom.
So actually, here the cursor is moving, sir. Here the black one is moving.
No, it's around 576 long database and big database. Okay. Anyway, now you go to data. Phylogenetic analysis.
Yes, sir. It's a nucleotide. So I'm giving it clear.
It's not a protein coding. Go back. Are you creating cancel? Phylogenetic analysis. It's not a protein coding nucleoside, okay?
Yes, sir.
It's an ITS, so it doesn't code for any. Now you say no.
Yes, sir. Yes, sir. And already it's activate only, sir. So I'm going to give yes.
Cancel.
Again, we are going for phylogenetic menu.
Yes.
And then here I'm giving the same parameter. So finally, it brings all the sequence here. So here, if you want, we can add our root trees. But as usual, in my version, it's changing the values here instead of giving a scale. Then we can go for a save current session and to I have saved this session. And for copying this image, I'm copying the image into clipboard. I'm going for a port 5. So it's very small, so I'm going to drag for you. Is it okay, sir?
Yeah, should be okay. I mean, not a good practice.
Yes. Then for getting caption, as usual, we will go for a caption menu and you can get a display caption in new window and copy this to clipboard. And then coming to my document, I'm pasting here. So to save this, as usual, saving to desktop. That's it, it's saved.
Thank you, Kupura. Thank you very much. It was very good.
Thank you, sir. Thank you, sir.
Can you stop it? Can you please stop presenting?
Yes, sir. Yes, sir. Yes, sir. Sure.
Okay, ladies and gentlemen, how many of you have now understood it? Please raise your hand. Now here is your assignment now. Listen. Please lower your head. How many of you did not understood it now? How many of you not understood it completely? Please raise your hand. Monica, you didn't understand?
No, sir, that was by mistake. Sorry.
Mahesh, you didn't understand?
No, I understood.
No, not Monica. Mahesh. Mahesh?
No, sir, because I think I'm using the.
Okay. Got it. Okay, so now, ladies and gentlemen, learning curve is not, it's kind of steep. What you do now? Go to Google. I mean, I'm not sharing my slide. Present screen. Wait. First, you go to YouTube channel, MycoAsia. No. Okay, here are some videos. Okay, couple of things you could do. So this is the video. Please watch this video. Okay, fine. Please watch this video. okay, of this video. Also watch today's session. We also watch the video of today's session. That will be uploaded to our YouTube channel. Be uploaded. Then, after watching these two videos, select one sequence of your interest. From NCBI GenBank or your own or newly generated sequence from your lab and construct of NGA tree, neighbor joining tree. Submit the tree file. Submit a Word file. I think that submit the tree file is enough. I'm talking about this MTX file, I think tree session file. I'm talking about MTSX file. MTSX file. Save the session file. Email to contact@MycoAsia. If it is good enough, we will send a certificate of participation. A certificate of participation. Is that clear? Please raise your hand.
Hello, sir.
Yes.
Can I make a recommendation?
Yes.
What you just sent now, can we also have it in the MycoAsia platform? What you just sent now for the certificate, the procedure for the certificate, can we have it there?
Sure.
Because the moment we all log out of this platform, it will be difficult to get the message.
Oh, wait. I will share the link to WhatsApp group. I mean, all may not be in the WhatsApp group.
Okay. Thank you.
No problem. If you are interested, you can join our WhatsApp group. Okay. And if you are interested, you can submit your details. If you are interested, you can give your details for MycoAsia network. I hope you can see it. Romana, can you copy these things?
Yes, sir.
Please lower your hands. Thank you. Yeah. Any questions now? You may please ask me. We're gonna wind up at 12:55. We have around around 7 minutes for discussion. If you have any questions, please do ask me now. No questions?
No questions. Sir,
Yes, Deepak.
sir, distance metrics are discrete data, which is better for the phylogenetic.
See, if you are just interested, I think can you mute yourself? Thank you. If you are interested in just basic identification, even the NJ is okay. But if you are thinking about describing a new species or reorganizing a group of fungi or organisms, some organisms, better to do maximum likelihood and Bayesian. That would be my recommendation, Deepak. Unmute yourself, please.
Okay, sir. Thank you so much.
And please mute. If you are interested in, am I audible? If you are interested in advanced topics in phylogenetic analysis, you can actually watch this video by Raza for advanced topics. Any questions, please?
So Sayali is having one question.
How can we done with phylogeny sequencing of the pieces? Oh, no, I'm not going to explain all those things. I'm here to just tell you how we can analyze newly generated sequence. If you want to know about how to process your PCR product and all those stuff, you may have to Google and find out from the YouTube. I'm not going to explain right now. Maybe we can have one more event, one more training program on that. I know it is very important to understand, to learn how to process your raw sequence data files, but that would be a different online event, Sayali. Is that okay? Thank you. Any more questions? You can send feedback about today's presentation or today's for feedback. Feedback can be sent to at MycoAsia.
Excuse me, sir.
Yes. Please.
So this is Pratika. I would like to really thank you because for us beginners, the words like data analysis or bioinformatics can be really frightening sometimes. And to use those tools can be a very difficult task. So thank you for explaining this concept in a very, very lucid way. And right from the downloading the software and everything. So really thankful to you for this lecture.
Thank you, Pratika. I mean, MycoAsia network is for all the young people who are interested to learn about fungi, fungal identification, fungal taxonomy, maybe fungal biotechnology. So we are always here for any kind of help that you might need. Thank you for joining in today, Pratika. Thank you very much.
Thank you, sir. Thank you.
And please visit our website, the event section. We update it with the new events, and the past events are also mentioned in the list. Thank you. Any more questions or anything? Please keep in touch. You can join our WhatsApp group. I mean, it's a very hyperactive group. Some people may not like it, but it's up to you. but you can always email us, or we are also available on social media like Facebook, Twitter, LinkedIn, Instagram. Okay, keep in touch. And for any queries, you can email us to at contact@mycoasia.org. Ramana, what do you think?
So, good afternoon, everyone. I thank Shenoy sir for introducing us to the Mega software and focusing on steps to convert our data into a phylogenetic tree. Thank you, sir, for being so interactive throughout the session. I also thank all the participants for being mutually connected throughout the session. And lastly, I thank Gunjan ma'am and Akshata for their kind cooperation. Thank you, one and all.
Thank you, Romana, and thank you, Dr. Shenoy.
Thank you all. Have a good day there. Bye-bye.